study isolates indicated that the CAMP45S promoter is also functioning in some animal cells, although the extensive biological, serological, and molecular while this promoter has low activity compared to canonical animals, particular strains of the total efficacy are significant. This observation suggests that the 35S promoter may have potential to worsen animals. The promoter is named CAMP35S serological identification. Tradition 35S chief strains of PBY are recognized. Because the coefficient of sedimentation of the virus transfer, whose expression is naturally driven by this promoter, originating by S potato virus C, is one of the most widely used, recognized, and was identified in the 1930s. It was discovered at the PBYC induces hypersensitive responses rather wide at the Rockefeller range of potato cultivars. These reactions include the formation of mild mosaic patterns or structured 600 nucleotide. Unlike the other strains of PBY, some PBYC strains are non-aphid transmissible. Genomic maps of CAM studies by Visser et al. 7 type 9 did not identify any of the local isolates as being PBYC, but it has the mechanism been reported to occur in South Africa. Is unique 10, 11, a second strain of PBY as PBYN translation. This strain of was described in tobacco plants growing close to potato plants. 13, a process that PBYN results in only happens on bacterial mRNAs, and mild or even no damage to the tuber. Depends on its association the ordinary strain of PBY is denoted as PBYO. Factor EIF3. Infection of a potato plant with the PBYO strain results in mild tuber damage and does not cause leaf. Necrosis. Insect transmission. Both PBYN and PBYO or aphid transmissible and occur in South Africa. In Europe, these two strains have been shown to have recombined to form PBYNTN 15, 16, or 5B. The PBYNTN has been accredited with the ability to induce potato tuber necrotic ring spot disease (PTNRD) 15 tubers. Damage by PTNRD become unmarketable and infection by PBYNTN thus results in a larger economic impact and infection by the other body form potato virus Y transmission PBY may be transmitted to potato plants through grafting, plant sap inoculation and through aphid transmission. The most common manner of PBY infection of plant material in the field is through the insect, and although aphids on their own can directly damage potato plants, it is the role as viral vectors which has been shown to interact the greatest economic impact. CAMV proteins, such as P2 and in cold climates, suggesting that it may also contribute in some degree to viral wingless aphids assembly and aphid-mediated transmission. In addition, P6 has been shown to bind to P7. Crops serve as breeding grounds for these aphids and form a temporary area of colonization before the aphids migrate to the potato field. P6 involves modification in moderate climates, such as in South Africa, aphids are thought to reproduce asexually on wheat, other crops, indigenous plants, and garden regulator of salicylic acid means that there are a number of aphids present year-round. The importance and effective and is most stringent monitoring of aphid populations is stressed and repeated by Radcliffe and Ragsdale in 2002 serves to inhibit as PBY virions are introduced to potato fields almost solely by winged aphids from the virus outside these fields. The nucleus aphids have not yet been linked to the spread of PBY to potato transcription fields. Because active 1 Nepalese root is required for accumulation, has been found to be most effective in its role as viral vector 517-21. but others such as Aphis fabae, Aphis gossypi,
to the
Aphis
nucleus.
nastorti,
Regulation of yaw-dependent signaling
Macrocypha
is
mucorbi,
cytoplasmic.
Mysis,
In nature, it involves the COI1
Mysis,
pathway.
Thoradon,
In contrast to
Humuli,
that of
and
SAW,
Rhopalocypha.
yaw-dependent signaling
Insertum
is
are also
increased
strongly
in the presence
associated
of
with viral
modified
transmission.
1-Nepalese-rupee.
17,
Replication
21, the
diagram
agricultural
depicting the
research.
steps in the
Council
genome
Vegetable
replication
and
of
Ornamental
cauliflower
Plant
mosaic
Institute,
virus,
ARC VOPI, 6 of
DNA
South
is
Africa
depicted in
identified
human
20.
RNA, including
5 species of
the
aphid
tRNA
able to
is
function as
depicted in
PBY
red. See text
vectors.
for more
22.
details.
The efficiencies of some of these aphids
by reverse
to function as
transcription.
PBY vectors
Viral
were also
particles enter a
established,
plant cell
Ragsdale et al.
unencapsulated.
2001,
At this stage, the
and were
viral
found to vary
DNA consists
between
of
the
3
different
fragments,
species.
one on the
In South
strand,
Africa,
alpha,
Aphis
and 2
fabae,
on the strand,
Aphis
beta,
gossypi,
and
and
gamma,
Aphis nastorti
which are
are
imperfectly
the
assembled
most
into a
common and
circular
efficient
genome with
PBY
3 gaps or
vectors found in the field.
discontinuities, D1,
5.
D2.
Apart from being classed
In
according
D3, the
to
viral
efficiency as
DNA
vectors,
enters the nucleus
aphids
where the
can also be divided
discontinuities are
into 2
filled in.
subgroups, namely
At this
colonizing
point,
and
the viral DNA also
non-colonizing
associates
species.
with host histones,
Colonizing
forming a
aphids
mini
are aphids
chromosome,
which
not
reproduce
shown.
and establish
The
themselves
host
on potato
DNA-dependent
plants,
RNA
specifically,
polymerase
while
transcribes from
non-colonizing
the
aphids
35S
do not
promoter
reproduce
all the
nor
way around the
establish
viral
colonies
genome.
on potato
Surpassing
plants.
the 35S
Colonizing
promoter.
aphids are better
This
adapted
creates
to
2
life on
copies of the
potato plants and
35S
are thus
promoter in the
generally
resulting RNA.
considered as better PBY
Transcription
vectors
also
than
initiates at the
non-colonizing
19S
aphids.
promoter, not shown.
Non-colonizing
The
aphids
viral
do not
RNAs
primarily
pass into the host
feed on potato plants,
cytoplasm
but do
where they are
occasionally feed on
transcribed.
them while
The
searching for a
3'
more
end of
suitable
the
host.
tRNF
Their
meta-nears to
lower
a site
efficiency as
corresponding
PBY vector
to
is cancelled out by the
discontinuity
sheer
1,
numbers
D1,
in which
near
they
the 5' end of the
occur.
35S RNA.
1923.
The
Because of
tRNFs
this,
met
all
high
aphids
synthesis
exist by
viral reverse transcription encoded by
as possible
the
vectors
new
and
alpha
numbers
strand.
carefully monitored.
RNase
Transmission of
removes the
PBY
RNA.
by aphids
The
occurs in
DNA-RNA
a non-persistent,
behind the
non-circulated manner, which
This
suggests
new
a less
DNA
intimate
finds the
interaction
35S
between
promoter
variants
at the 3'
and
end
vector than is the
of the
case of
RNA
circulated
template. The
virions.
synthesis of the alpha
24.
strand of
The
DNA
fact that the
continues and
virions are
RNase
transmitted
H continues to
in a non-persistent
degrade
fashion
RNA
means that
complex
viral
to
replication
DNA.
does not occur
Synthesis
within
of
the
the
aphid
alpha
vector
strand
and
completes.
that,
RNase
unless the
H
aphid
activity
feeds on
exposes
infected plants, it
purine
loses
rich
its
regions
ability
at the
to
position
infect
of
plants after
discontinuity
2-2.
3,
Three
which
feedings.
primes the synthesis of the gamma.
525. The virions
DNA
attach to the aphid
RNase
stylet
H
in a matter of
activity
seconds
exposes
and may
purine-rich
remain
regions at the position of
infectious for 4 to
discontinuity
17 hours.
2,
26.
which primes
27.
the synthesis
The
of the
distance
beta
over which the
DNA strand.
virions can be
When the new gamma strand of
transmitted is
DNA
limited
reaches
due
the
to the
5'
short
end
period for which they
of
remain
the new alpha
infectious.
strand, it switches
23.
to the 5'
Although
end
the
of the new alpha
short
strand,
lifespan
recreating
outside plants
discontinuity
inhibits
1.
long-distance viral
D1.
transmission.
When the
It
new
does
gamma
not reduce
strand
the
of DNA reaches the
transmission
5' end
efficiency
of the new
bestowed
beta
by the
strand,
quick
it
rate of viral
displaces
acquisition and
the primer
inoculation
and some of
within
the
a
newly synthesized
field.
beta strand,
Upon entrance
resulting in
into
the
the plant
recreation
cell,
of
the virus
discontinuity
coat
2.
protein disassembles and
D2.
releases
When the
its
new
RNA.
beta strand of DNA
Genome.
reaches the
The
5'
viral
end of the new
RNA
gamma
serves
strand,
as
it
mRNA,
displaces
and
the
although little is known
primer
about the
and some of the
translation
newly
thereof,
synthesized gamma
it
strand,
is believed
resulting
that
in
the
the
5'
recreation of
non-coding
discontinuity
region functions as an enhancer
D3.
of translation.
At this point, the
28.
new viral
The
genome can either be
translated
packaged into
mRNA
capsids and
results in a
released from the
polyprotein which is
cell, or
processed
they can
into
be
mature
transported
proteins.
by movement proteins
Each
into an
polyprotein
adjacent, uninfected
is then
cell.
cleaved into 10
14.
different proteins which are believed to be
Cauliflower mosaic
multifunctional.
virus promoter
These
CAMV35S
along with host
is used
proteins,
in most
assemble to
transgenic
form a
crops to
replication
activate foreign
complex.
genes
This
which have been
complex
artificially
performs
inserted into the
negative
host
strand
plant.
RNA
It is
synthesis
inserted into
using the
transgenic
positive strand of
plants in a
viral
form which is
RNA as a
different from
template.
that found when it is
Once
present in
the
its natural
additional
brassica.
RNA copies
Plant
have been
hosts.
produced,
This
they code
enables
for
it
the
to operate
synthesis
in a
of
wide
various
range of
proteins
host
as
organism environments which
mentioned
would
before, as well
otherwise
as
not be
proteins.
possible.
These
CAMV
proteins will
contains
now
about 8
enclose the
kilobits
newly
double-strand
formed
DNA
genomes to give rise to new virions.
Spherical
It has been
particles.
suggested that enclosure of the newly
infections are
formed
systemic,
virions is
DNA
initiated by the
infectious
interaction of the
inoculated.
coat proteins with
On
the 5'
abraded plant
terminus and that
surfaces,
the
the CAMV
protein is
genome
built
has
up
8
towards
tightly
the 3'
packed genes,
terminus,
of which only 2
29.
small
The entire process of
genes 2 and
viral
7
replication
are non-essential.
occurs
As a
within
result,
the
only these
endoplasmic
two genes
reticulum.
can be
These newly
replaced,
synthesized
deleted
viral
without a loss
particles
of
are subsequently
infectivity. In
transported
addition,
through the
modified
plasmodesmata
CAMV
to
genomes
adjacent plant
exceeding
cells
the
via several
natural
assisting
genome size
polyvirus
8024bp
proteins.
by even a few hundred
Distribution of
bp are not
viruses within the
packaged
plant
into
occurs
virions.
according to the
These
source-sink
two
relationship
factors seriously
between
limit the
maturing
size
and growing
of DNA
tissues.
insert clonable
30.
in CAMV.
Virus concentration
The
throughout the plant
bacterial
is high, and this
dihydrofolate reductase
greatly increases the
DHFR
chance
gene
of
has
uptake
been
by
successfully cloned into the
infection of plants by
CAMV
polyviruses
gene
can be
2
varied. In the symptoms
been
shown,
successfully
infections
expressed in
include
plants.
anal necrosis,
Molecular
mosaic
mechanisms of
symptoms as well as
vector-mediated
leak,
CAMV
malformation.
transmission. The
Ulam
virus
et
is
al.
acquired from an infected
2002.
host during feeding by
Infected
the
plants
aphid
that do
vector.
not show symptoms may have infected.
A
Cannabis
transmissible
will yield
complex
lower
is
quality
composed of
products than
virions
the
and
healthy
protein
counterparts.
located in the
Potato,
vectors.
PBYNTN.
The P2N
Interaction
terminal domain
since
recognizes a protein
PBYNTN causes
receptor
great loss
located
in
at the tip
potato
of the
production.
stylus.
The research of
In the
potato,
P2C terminal domain binds to
virus
the PE
YNTN
decorated
interaction is
virions.
important. Sensitive
Transmissible
potato
complex
cultivars respond to
CAMV
PBYNTN inoculation
is
with
controlled by the tissue
development
and
of
intracellular
typical symptoms.
specific
On inoculated
localization
leaves
of
5 to
P2.
7 days
This
after
protein is only
inoculation,
found in
parotid
epidermis and parenchyma
necrotic green
cells.
spots develop.
Moreover,
As the virus
these
spreads
cells,
through the plant,
P2
the
is
systemic
localized in
symptoms
single
develop
viral
on
electron-loosened
uninoculated
inclusion
leaves.
bodies,
10 days after inoculation, wrinkles and mosaic fluorosis appear, leading to a
first produced in
punctured
numerous
appearance,
viral
leaf
factories,
drop. The
electron-dense
viral defense
inclusion
mechanisms of plants will
and are
primarily
later
try to
exported and
restrict
co-localized
the
with
movement of the virus.
microtubules before
In failing
concentrating
this,
in
it may
ELIV.
attempt to induce cell death,
CAMV specifically
infected
uses the
tissue,
microtubules
thereby
to form
preventing
the
the spread of
transmissible body
virions.
and thus enable
31.
vector
Although the precise
transmission.
mechanism
The
of
complete molecular
disease induction by
characterization and study of this
polyviruses in
virus
plants
was
is
not
unknown.
known.
It is known that these viruses
Evasion
cause
of
a
plant defenses
significant
cauliflower
shutdown of
mosaic
host
virus
gene
possesses a
expression
number of
during viral
mechanisms that
replication.
allow it to
32,
counteract
33,
host plant cell
34,
defenses,
physiological.
while the pregenomic
Changes in
35
potato
sRNA
plants
is
as a
responsible
response to
for
PBYNTN
genome
infection were
replication by reverse
intensively studied.
transcriptase. It also
Early
contains a
stages
non-coding
of infection,
600 base
meaning
pair
first
leader
12 hours,
sequence that serves
photosynthesis
as an
related
important
genes
mRNA
genes
for the
involved
production
in
of factors involved in
perception,
viral
signaling and
counter
defense
defense.
response were
A
shown to
number
be
of hosts of
differentially expressed.
CAMV possess
34,
small RNA-based
24
viral
hours.
silencing
After
mechanisms
inoculation, the amount of
to limit
salicylic
viral
acid
infection.
increased.
The products of the
35,
aforementioned
a disruption
600
in gene
BP
expression
sequence are viral small
disrupts the normal cellular
RNAs,
function of cells, which
vsRNA,
could be the
of
cause of
21,
the physical
22,
symptoms
and 24
that the plant
nucleotides in length
demonstrates.
that serve
At
as
the time of
decoys,
symptoms
binding and
development,
inactivating.
research on interaction
Effectors
between
of host silencing
susceptible
machinery,
potato
such as
cultivar and
Argonaut-1,
PBYNTN
AGO1,
showed changes in
as proof
cytokinin
of
level.
principle.
36,
Experimental
in inoculated
overexpression of
leaves,
these VSRNs
showing
allows for
symptoms
increased viral
modifications in
accumulation in
chloroplast structure
infected
and size.
plants.
37,
Concerns
lower
about use
chlorophyll
of
levels and
CAMV35S
differential
promoter in
activity of
transgenic
soluble and
plants in the early
ionically bound
2010s.
peroxidases. 38 were
Concerns have
detected.
been raised about using
At
the
later stages of
CAMV35S promoter for
PBYNTN
expression in
infection, total
transgenic
protein
plants.
concentration
Because
increased in
sequence
sensitive
overlap
potato
exists between
cultivar
this
while
promoter and the coding
no such
sequences
pronounced
of P6.
changes were observed in tolerant
54.
and moderately tolerant
Transgenic
potato
events certified
cultivars.
for release
39.
in the USA contain up
Gene
to
expression
528
studies revealed
BP of
changes in
ORF-B
expression of
encoding.
genes for heat
See
shock
terminal
proteins,
domains
catalase.
of P6.
Beta
As
minus
P6
1,
is a multifunctional
3 glucanase
protein
and
whose
genes
full range
involved
of
in
functions
photosynthesis.
is
33.
unknown. There is some
Molecular
concern that
description of
expression
potato
of one or
virus.
more of its domains
Why
may have
polyvirus
unforeseen
virions consist of
consequences in the
non-enveloped filamentous
transgenic
structures
organisms.
that are
Recent
680 to
studies have
900
attempted to
nanometers
determine
in
what length of
length and 11 to 15
CAMV35S
nanometers in
promoter
width.
has the least chance
40.
of inadvertently
Morphologically,
producing
the
P6
polyvirus
domains
capsid
while still
consists of
retaining
approximately
full promoter
2,000
activity.
copies
As
of
one
coat
might
protein,
expect, using shorter
CP30.
promoter
The
lengths
capsid
decreases the number
encapsulates
of
a single strand of
P6
positive
domains
sense
included and also
RNA,
decreases
which is
the
in the order
likelihood
of
of
10
unwanted
kilobits
effects.
in length and has a non-translated 5-terminal region. 5. NTR, as well as a 3. Poly a tail. 41, 42. The positive sense genome contains a single
extended open reading frame and acts directly as mRNA. The 144 nucleotide 5, NTR, is particularly rich in adenine residues and has very few guanine residues, rather than a conventional cap structure. The 5' NTR is associated with a viral genome-linked protein, VPG, which is said to act as an enhancer of transcription. 28. The 5' sequence has an internal ribosome entry site, IRIS, and cap-independent translation regulatory elements, CIREs. 43. The IRIS directs cap-independent translation through a mechanism similar to that used by eukaryotes. 44. The extended open reading frame encodes for a 350 kDa polyprotein. This polyprotein is proteolytically processed by viral proteases, NIA, HC-Pro and P1, and undergoes co- and post-translational cleavage to yield several multifunctional proteins. These include the following: P1 protein, HC-Pro, helper component proteinase, P3 protein, 6K1, 6kDa protein 1, C, cylindrical inclusion, 6 kelvins 2, 6kDa protein 2, VPG, viral protein genome linked, NIA-Pro, nuclear inclusion protein A, proteinase domain, NIB, nuclear inclusion protein B, and the CP, coat protein, 30, diagnostic techniques for detection of potato virus Y. ELISA. In the past, crops were inspected visually to determine whether or not they were disease-free. Visual inspection was also used as a basis for seed certification. Determination of viral status through visual inspection is incredibly difficult, as the symptoms may be masked or the infection latent. 23. As a result, post-season tests and inspections were introduced. These tests involved the cultivation of previously harvested material in greenhouses. The resulting plants were inspected for a more accurate estimate of viral status. Although this method of screening did offer some degree of monitoring of viral presence, it was subjective and highly ineffective. Enzyme-linked immunosorbent assay, ELISA, screening of crops and seed potatoes replaced visual inspection in the early 1970s. The use of ELISA offered routine diagnostic laboratories a quick, effective and sensitive method of screening for a wide range of potato plant viruses. Detection of pathogens using ELISA relies on the interaction between the antigen and specific antibodies and has become a popular and cost-effective means of routine detection. In an ELISA, the solid phase can be coded with the sample of interest containing the antigen. 45. The efficiency to which the antigen binds to the solid phase is dependent on temperature, length of exposure as well as concentration. 45. Solid phases used include nitrocellulose membranes, paper, glass, agarose and polystyrene or polyvinyl chloride microtiter plates. Microtiter plates are the most widely used solid phase because they are easy to handle, allow for automation and for analysis using microtiter plate readers. A drawback of these plates is that they are highly absorptive, and this increases the incidence of nonspecific binding of components used in the ELISA. Nonspecific binding to the plates is reduced through the use of buffers containing proteins such as casein and non-ionic detergents such as Tween 20. After coating, excess sample is removed and the plate typically treated with a 1% casein-containing solution. Subsequent to this, the solid phase is treated with antibodies raised against the antigen of interest. After each incubation step, the plate is washed with Tween 20 containing PBS. These washing steps are aimed to wash away any non-specifically bound components. 46. Non-specifically Bound components are less strongly bound than the specific bound ones. Detection is achieved either through the addition of an enzyme-coupled antibody or the addition and detection of a biotinylated antibody. In a system using an enzyme-coupled antibody, the subsequent addition of an appropriate substrate results in the formation of a color proportional to the amount of antigen. 46. Alternatively, the plate can be coated with antibody followed by incubation with the sample that is to be detected. This, in turn, can be detected as described above and is then referred to as the double antibody sandwich, DAS, ELISA. Both of these systems, however, have a disadvantage in that coupling of the enzyme to the antibody may result in steric hindrance, which in turn may result in a loss in function of the antibody and/or the enzyme. 47. This may be overcome through the use of a biotin-avidin or biotin-streptavidin bridge. In this type of system, biotin is coupled to the antibody. The biotin molecule has no influence on the working of the antibodies and is easily detected using avidin or streptavidin conjugated to a suitable enzyme. Streptavidin has an extremely high affinity for biotin, which results in even a higher degree of specificity than a system in which the enzyme is coupled directly the antigen. To establish whether or not the antigen is present, a substrate specific for the enzyme used is added. The enzyme then converts the substrate to a colored product, and the color intensity can be correlated to the amount of antibodies bound, and thus the amount of antigen present. A DAS ELISA has the advantage that it can increase the specificity of the ELISA and reduce the occurrence of nonspecific binding. As a result, the DAS ELISA principle is commonly employed in ELISAs for the detection of plant pathogens in plant sap without prior purification of the pathogen. The ELISA is considered to be a safe, inexpensive, and rapid method for detection of plant viruses. The inexpensive nature and relative simplicity thereof allows for it to be used as a workhorse within the agricultural sector and is used to screen thousands of samples per year. Unfortunately, ELISAs are not completely failsafe. Virus levels within potato tubers, which are screened by ELISA for use as seed potatoes, are normally low, while the tubers are dormant. ELISA detection of viruses in these potatoes is difficult and absorbance values may fall below the set cutoff value. For this reason, seed tuber screening is performed on sprouting rather than dormant tubers. Although this results in more reliable readings than direct tuber testing, it does delay the certification of seed potatoes. 48. Another disadvantage of an immuno-based detection method is that changes at the gene level may have an influence on the immunogenicity of the antigen to be detected. In terms of potato plant viruses, mutations within the CP gene may cause the CP to undergo conformational changes, rendering antibodies produced against the previously present virus less effective. RT-PCR reverse transcriptase polymerase chain reaction, RT-PCR, has become a powerful and effective method for detection of potato plant viruses within potato plant material and even dormant potatoes. Only a minute piece of plant material is required for analysis using RT-PCR. Considering the protocol described within this thesis, 0.1 grams of plant material is enough for 14,500 separate reactions. During a RT-PCR, specific target RNA sequences are amplified exponentially into DNA copies. For this to occur, however, the RNA of the virus must first be transcribed to DNA by means of a reverse transcriptase polymerase. This polymerase synthesizes a DNA strand using the RNA as template. This results in a DNA-RNA complex. For synthesis of a DNA strand from the RNA template, only the reverse primer is required, since the RNA is a single strand arranged from 5' to 3. Subsequently, the newly synthesized DNA strand is used as a template for traditional PCR. Different types of reverse transcriptase polymerases are available to suit different needs and reaction conditions. Reverse transcriptase enzymes commonly used include AMVRT, superscript 3, IMPROM2, Omniscript, Sensascript, and TTHRT. At the end of the RT step, the polymerase enzyme is heat activated. It could also be that the reverse transcriptase polymerase and DNA polymerase is one and the same enzyme, and that the enzyme only requires a DNA polymerase activation step after the RT step. An example of such an enzyme is TTH polymerase. This enzyme has both RNA-dependent reverse transcriptase and DNA-dependent polymerase activity. However, the active center of the DNA polymerase is covered by dedicated oligonucleotides, called AP tamers, at temperatures below the optimal reaction temperature of the DNA-dependent polymerase component of TTH remains covered by the AP tamers. At these temperatures, the TTH enzyme only synthesizes a DNA copy of the RNA template. Once the reaction temperature is raised to 95 degrees Celsius, the AP tamers are removed and the DNA-dependent polymerase component will start to amplify the target sequence. PCR amplification of the DNA target occurs in 3 steps: denaturation, annealing, and extension. 46. Each of these steps occur at a specific temperature for a fixed period of time. Denaturation is normally allowed to occur between 90 and 95 degrees C and leads to the dissociation of DNA strands. After this, the reaction is cooled to between 40 and 70 degrees Celsius to allow the primers to associate with their respective target sequences. This step is known as the annealing step and is primer-specific. The temperature at which the primers anneal is critical. Too high temperatures would not allow the primers to associate with the DNA, resulting in no or poor amplification. Too low annealing temperature would ultimately lead to nonspecific binding of the primers and nonspecific amplification. 46. Primers bound to the regions flanking the target DNA provide 3 hydroxyl groups for DNA polymerase catalyzed extension. The most commonly used DNA polymerase is TAC, a thermostable enzyme isolated from the thermophilic bacterium, Thermus aquaticus. The DNA polymerase synthesizes new DNA strands along the template strands, using the primers as starting points. During the extension step, the strands are amplified beyond the target DNA. This means that each newly synthesized strand of DNA will have a region complementary to a primer. There is an exponential increase in the amount of DNA produced as the 3 above-mentioned steps are repeated in a cyclic fashion. In a traditional PCR, these steps might be repeated 20 to 55 times. A problem, however, with PCR amplification is that the temperature required for DNA strand dissociation also results in DNA polymerase denaturation. This is partially overcome by the bioengineering of polymerases, which are more thermal stable and have longer half-lives. Even though RT-PCR is technically more difficult to perform and more expensive than ELISA, it has the ability to allow for the detection of low viral loads. RT-PCR is considered to be 102 to 105-fold more sensitive than traditional ELISA. 49. RT-PCR also allows for the detection of several viral targets in the same reaction through the use of several primer combinations. This is called multiplexing. Although multiplexing is technically more demanding than a traditional simplex reaction, it allows for a higher throughput in that a single sample can be tested for several viral strains in a single reaction. Primers used for multiplexing are chosen in such a manner that they result in amplicons of various sizes. This allows for post-RT-PCR analysis using gel electrophoresis. Although RT-PCR saves time, allows for multiplexing, and is more sensitive than ELISA, the reagents and instrumentation needed are expensive and require a higher level of technical expertise. Also, end product analysis using gel electrophoresis is laborious, relatively more expensive, time-consuming, and does not lend itself to automation. For these reasons, the use of RT-PCR for routine screening is not feasible and has not replaced ELISA. It does, however, provide the industry with the opportunity to screen borderline cases, especially in the case of seed potato certification. Quantitative PCR. In most traditional PCRs, the resulting products are analyzed after the PCR has been completed. This is called endpoint analysis and is normally qualitative of nature rather than being quantitative. For this sort of analysis, products are mostly analyzed on an agarose gel and visualized using ethidium bromide as a fluorescent dye. Direct correlation between signal strength and initial sample concentration is not possible using endpoint analysis, since PCR efficiency decreases as the reaction nears the plateau phase. Quantitative PCR, however, offers an accurate and rapid alternative to traditional PCR. Quantitative PCR offers the researcher the opportunity to amplify and analyze the product in a single tube using fluorescent dyes. This is known as homogeneous PCR. During a quantitative PCR, the increase in fluorescence is correlated with the increase in product. Through the use of different specific dyes, quantitative PCR can be used to distinguish between different strains of a virus and even to detect point mutations. The major advantage of quantitative PCR is that analysis of resulting products using gel electrophoresis is not required. This means that quantitative PCR can be implemented as a high-throughput technique for sample screening. Quantitative PCR has been described for detection, 50, and discrimination of PVYO and PVYN isolates, 51, 52, and for reliable discrimination between PVYNTN and PVYN isolates, 53.